Hi everyone, welcome back. In this video and in the next few videos, I'm going to focus a bit on biological applications, of course using image analysis and of course using Python to automate this image analysis process. You know me, I love Python. This is all this channel is about. And by the way, if you haven't subscribed yet, this would be a great time to pause and hit the subscribe button. You know where to find it, so I don't have to point out. So I guess right there. Okay, now this video is about Sholl analysis, and if you are a biologist, you probably know what I'm talking about. If you are a neuroscientist, you definitely know what I'm talking about. And if you are not any of these, I think it's still useful for you to watch the video. For example, if you completely do something else, you may be working on fiber analysis. Maybe you learn something about how to adapt what we are trying to do for your specific analysis. There's always something to learn from any of these videos. Again, this is focused on using Python to kind of automate these processes. Okay, there's a lot of talking, but let me go ahead and jump into actually defining what Sholl analysis is and then jump into Python code. Okay, so first of all, what is Sholl analysis? It's an assay. Again, in biology, a workflow, typically you call that an assay that is measured to perform how these neurons and dendrites or axons, not just neurons, basically the neuron dendrites or axons, how they branch out from the cell body. Okay, so it's a way to quantify this. Now, why would anyone do this? Why would a neuroscientist work with this type of analysis? Again, there are many other reasons. The couple of things that I most often encounter during my discussions with researchers is one, to compare the neuron morphology between different cell types or between different conditions. The other reason is, of course, to assess the changes in the structure of these neurons during the development of a disease. During the development process or the evolution of a disease or during a disease, basically, mixing my terms here. So these are the couple of reasons. Now, how is it performed? Well, a typical workflow includes putting a series of concentric circles on an image of a neuron. You just put like a whole bunch of concentric circles of fixed width around a neuron. Now, where do you center that? You center that around the cell body, which is called Soma, right? So this is the brightest thing usually, and you put that in your image, or the biggest, brightest thing. You put the center on it and you put a series of concentric circles. And then you count the number of times the neuronal process, intersects, like how many times these neurons actually, dendrites, they intersect each circle. And then you count how many of those, and then you plot them. So this kind of gives you, in a way, an idea of how the neurons actually, the structure is out there. Now, the question is, this used to be completely manual and still is manual for many people. In fact, there is a semi-automated way of doing this, for example, using ImageJ. So if you're not a Python coder, I definitely encourage you to use the plugins that are available in ImageJ. But obviously, you're watching my channel, this video, to learn how to do this in Python. So please stick around. So how to pick Soma for Python-based analysis? This is the next question. So obviously, when you're doing it in a manual way, you're picking everything manually. But when you want to automate this task, one way to do this is you assume that the Soma is the brightest pixel in your image. And that is a horrible assumption, as you'll probably see when we get to the code, because oftentimes you may have some other region in your image that's lighting up a light bulb for whatever reason that has the brightest pixel. And in an automated way, that would be the center, but that's probably not the Soma, right? So that's why you kind of need a semi-manual way of actually performing this analysis. You can find some advanced tricks to find Soma. Not all of those are reproducible, meaning you send multiple images through your pipeline. You may not consistently get these results. In fact, you can actually combine deep learning with this analysis. Maybe I'll do a video on that in future, where you can consistently detect Soma kind of almost accurately. With deep learning, you can actually do that. I'm talking about traditional thresholding methods. It's near impossible, but with deep learning, you can get the location of Soma in an automated way. I don't want to combine deep learning with this. I've done enough videos on image, the object detection, for example, to classify Soma. Maybe I'll do one of those in upcoming videos. But for now, let's look at automated way of identifying this pixel. But more importantly, I want to do the manual way where an expert identifies where the true location is, and then the analysis continues. So that's our plan. And what if you want to perform the Sholl analysis on multiple images? Right now, what I'm going to show you is a single image. Hey, you have this dendrite structure, and here is the Soma. Go ahead and perform the Sholl analysis and give me the result. Now, if you want to perform that on multiple images, let's save that for next tutorial. Okay, but for now, let's focus on a single image, and let's go ahead and jump into the code. Okay, so here is my code. Again, I'm gonna share the file as usual. So if you want to take notes, fine, but you can just download this. I added all the notes, relevant notes in terms of what Sholl analysis is, and I'm pretty sure you know what it is. I also added a link to one of the test images that I've used, and of course, please add any in case, hopefully you're not using someone else's images in your publication. This is for just learning purposes. So, but here is the link if you want to download it and work on it. How did I find this? I just did a Google search on neurons and I found one, and that's what I'm ending up using right now. Okay, so I'm using Spider IDE, and I have a weird feeling that it decided to quit on me. You see how I can't get anything going. Let me restart this because it's been a while. I've been using it for various purposes. Let me quickly restart this and continue. Sorry about that. Okay, so I didn't have to restart. It actually fixed itself. I hope it stays that way. I haven't seen this IDE crash, but oh well, you never know. I'm actually having too many things open. Maybe I should close some of those. Okay, let's jump into the code right here. What are we trying to do? We are trying to first identify the Soma, right? The workflow, which is the center from which these circles are radiating out. So we need to first define that center, and then we need to define the circles, and we need to define the distance between these circles. I am working with an image that's not calibrated, so my distance is going to be in terms of pixels. Otherwise, you'd give that in terms of microns, right? So you do the pixels to micron conversion. So first, let's do the auto method. So there is identify Soma auto method here, and there is a manual Soma selection method down here. So as the name suggests, auto automatically identifies. And how is it identifying that? As you can see, I am using the moments right there. Actually, where is min-max? Right there, min-max location. All it does is it finds the maximum or the peak location of a given pixel, and we are actually identifying that specific pixel, and that's it. And it returns the coordinates right here of that specific pixel. So that is the auto way of identifying this. Of course, I'll show you how it looks like, but we'll immediately figure out, hey, this is not the Soma. Let's see. Okay, the next method here is manual Soma selection. As you can imagine, I'm using OpenCV, which means I can actually use our mouse callback right there, and then use our way of actually clicking on it, and then where it returns the coordinates. Yeah, so that's what we are using down here. And again, this is I don't want to go through line by line. I'm pretty sure most of you know OpenCV. If not, just use these. It's basically we are using a mouse callback, and we are actually using the left button down, which means whenever I click the left button down, it's going to extract these coordinates, and it's going to basically, again, close all the windows when we actually click Q, or you can destroy the window. Basically, if the window doesn't close, you can go ahead and close it, and then it captures these. Again, depending on your coding environment, this may or may not work, so you may have to update this. Again, I say this because on Google Colab, this probably doesn't work. This works down here on a local IDE like Spider or PyCharm, for example. So it destroys all the windows, meaning once I left click it, it gets the coordinates and then it closes the window. Okay, I didn't expect spending 2 minutes discussing that, but that's what's happening right there. and or when I type Q, it just exits the window, right? You can destroy the window manually that way. Okay, then it gets the Soma center. Again, this function basically, all I'm saying is, hey, is it auto or manual? Whatever it is, go ahead and give me the XY coordinates and return those coordinates. That's what this function is all about. And below here, we are just, this is the meat of it. This is the bulk of main code. And we are starting obviously by loading an image using OpenCV. And we don't care if they are grayscale. For example, in my case, I think, let me open my image right there. And I have two images right here. In both cases, of course, in most of the time, you may have fluorescence images that are in color. So you can see, or Soma, you can see in both images. But I am going to work with grayscale images. That's what I'm doing there. And then I'm converting that into a binary image by thresholding. Why? Because I want to work with a skeletonized image. So I have all these neurons, and each neuron, for example, let me open, yeah, this image. You see how this one is a bit thick, this one is a bit thin, this one is a bit thick again. So we want to collapse all of that into single pixel, which is what we call skeletonization. And this is where it requires a bit of experimentation again, because one setting may not work. You may have gaps in your neurons. So you need to play a little bit with your skeletonization. The best one that works for me is, skeletonized right here. Where are we importing that from? From scikit image morphology. There is a function called skeletonize, and that's exactly what I'm using. So I experimented a little bit, and this is what is working for me. So I hope it also works on your image. And yeah, go ahead and use these settings and see if the skeletonization works for you. I'm dwelling on this because skeletonization is an important component of this entire Sholl analysis. If you have breaks in your neurons, and if your thin neurons, if you're eroding your pixels and your thin neurons are gone and the thick ones are remaining, then obviously you're going to get bad results. So this is an important step, skeletonization. So make sure you get good binary image and good skeletonized image. Then, hey, you're getting Soma center, meaning where is my center of this, where is the Soma located? And from that, we define like what is the radius. Like in my case, I'm defining 20 pixels. That's the distance between these concentric circles. And then what is the maximum radius? How far out in your image do you want to go? I put 60%. I mean, initially I put 75%. I changed it to 60%. We'll see. I mean, we can change these and see how they look like. Obviously, initially I also tried, okay, my image radiating out is, look at the minimum of my image dot shape, which basically is what is the smallest dimension, and I want to go half of that as my max radius, which means it covers the entire image in that dimension. But I changed my mind, and I'm using this approach. It gives you a bit more control, right, in terms of what the radii, maximum radius should be. Okay, and then we define a few of these radii within these limits, and now we are going to display, for display purposes, I'm going to create an image with these colors overlaid. Again, we'll see that, and then actually we are doing the Sholl analysis down here. How are we doing that? Well, you create a disk right there, and you create a mask, and wherever there is an intersection between the mask and the actual neuron in your skeletonized image, we're going to call that an intersection, and we are going to count that as 1. That's it. It's as simple as that. You see, I'm using the logical AND between the skeleton and the circular mask. If the skeleton and the circular mask, if there is an intersection, then it counts. Otherwise, if it's a dark pixel, which means it's a background, then we don't have an intersection. It's as simple as that. Okay? And then we are counting how many non-zeros are there. That's it. And we are capturing that down here. And of course, at the end of it, we are actually plotting everything for visualization purposes. And how do you quantify this? Okay, now you count it. The most common thing that many of you do is how many times it intersected versus the distance, right? So this is the basic plot. But there are other ways, other things that you do. Sometimes it can be a semi-log plot, a logarithm on y-axis and just a linear scale on the x-axis, or log-log plot. And you try to look at the trends. Is it a straight line? And then if so, fit it to a straight line and give me the slope and give me all the other parameters that you want to extract. I am only doing a single fit right here. In fact, I called it linear fit, but we can fit it to degree of 3, for example, degree of 4, 5, whatever, to actually fit this curve. Okay, with that information, I think we are all set. I don't want this video to be any longer than it needs to be. So let's actually work with image. I think, which image do I have? Obviously, this is one image at a time. We'll do multiple images and aggregating the information in the next video, because aggregating information, what if one image is at a different scale? What if one neurons are larger than the other? So when you're comparing them, you need to have the radii to be exactly matching, and you need to combine the information accordingly. So let's quickly look at that in the next video. But for now, we are doing one image at a time. And we are looking at neurons3.png. So let me run the entire code. And the first thing it should do, well, actually, we are doing this in an automated way, right? So it's not going to ask me anything. You see on the screen, it says, hey, the Soma center is at 89 and 590. Is that correct? Obviously not. As you can see, let's go through some of these results. First thing, what you see here is the original image converted to grayscale. This one is the binary image. And this one is the skeletonized image. And if you go back to the original image, you see these faint areas here, faint areas. They're faint in the original image. In fact, if you only look at the skeletonized image, you're like, okay, did I do any mistake? You should go back and make sure it's not a mistake. It's actually present in your image. You can see how amazing this skeletonized image is here. So we are good. Oh, sorry, here. So we are good. Now, on the skeletonized image, we need to overlay these circles. That's what we're doing here, but centered around the Soma, which is obviously right here. It's not here. This is the problem with manual. It thinks, hey, automated. It thinks we said the brightest pixel, so it goes to the brightest pixel. This is where maybe identifying Soma using deep learning can be useful. But again, if the Soma has different sizes, for example, let's do, I don't know, do I have a neurons.jpg? Let's go ahead and run this. I want you to see how there is some limitation with a manual process. This is a different image. Oh, it actually did not do a bad job identifying the center. Oh, let's go back. Identifying the center of this Soma. If I can click on it, where is it? Right there. The actual one is right here. Still, the brightest pixel is very close by in this specific case, but it's not really exactly on it. And yeah, so this is the challenge when you have different images of different sizes. Okay, now let's go ahead and switch this from manual to auto. I think I defined that at, I should move all these inputs to one place, image and everything, but it's okay. Let's go ahead and change this to manual and run this exactly the same way one more time. Oh, the image showed up on my other screen. Let me drag it to this screen. So that's what it popped up, OpenCV, right? So now it's waiting for me to click on it. As soon as I do left click, it should exit the screen. It should take those center coordinates and it should redo this entire analysis. And now you will see, there you go. And now you will see all the results. So there is our input image, there is the binary image, skeletonized image, and there is my Soma manually provided. And I'm just this print I was looking at, is my skeletonization good? Is it over skeletonizing, under skeletonizing? So this is why I'm printing this, so I can do some troubleshooting. But at the end of it, it's actually showing me the number of intersections and the radius. That's what we're plotting at the end of it. And I'm also plotting as we went through, I'm trying to fit this to a degree 3. You can try degree 2 if you want. And you can try degree 4. The higher orders, the more complicated it may actually look like. But yeah, I changed it to 2. yeah, this is not really that great, right? So order 3 seemed to work okay here. So let's go ahead and fit this. And again, why are we fitting this? So we can actually print out the coefficients that you see down here. So let's see. This is the last, and we'll change the image and then look at how how this curve looks like for a different image. But you can see these are the coordinates. We have the r squared, AIC, and BIC, everything printed down here. Okay, so now let's just check this on. You see the manual is not that painful. You just still need to give where the center point is, but once it is provided, it should work very well. Okay, I think I also have neurons2.png, so let's go ahead and run it. And there you go. And let's go ahead and identify the Soma location and then let it do its job. And after it's done, in fact, let's also do the third image so you can see how things work in a very consistent way. So there you go. And here is a different curve. You may have to change the order for some of these, right? I mean, order 3 worked for the first one, but not for this one. But again, here is the goal. Okay, just to finish this off, let's just do neurons 3. This is my favorite image. It looks pretty cool. It'll pop up any second on the screen. There you go. This is a high resolution image, obviously, no need to mention. And let's wait for the result and meet again in the next tutorial once we have a quick look at the final result. There you go. Oh, order 3 works very well for this image, and skeletonization was good. Everything looks perfect. So this is Sholl Analysis. In summary, this is not a complicated essay in biology, but still a bit challenging to automate, as you can see. Okay, I hope you found this to be useful, informative, and again, if you have any questions, requests, leave that down in the comments. Do not forget to like these videos, and of course, do not forget to subscribe. Let's meet again in the next tutorial. Thank you very much.